All right, let's begin with the history of life and evolution notes. This first recording will just be the first half of the notes on the history of life. So let's begin. All right, here we go. Earth is about 4.5 billion years old. Basically, what they think happened is that there was this giant ball of gas and dust that started to spin and swirl. Over time, it started condensing and getting tighter and tighter, and eventually, what formed was our solar system. Towards the center, not quite exactly at the center of the solar system, is our sun, and basically, Earth happens to be the third planet from the sun. Radiometric dating. Why is this important? We're talking about history of life on Earth. Radiometric dating is important because this is the tool that we use to date how old a fossil is or a rock may happen to be. How do we do it? We look at isotopes and what happens to these isotopes over time. So let's first talk about what an isotope is. For example, hydrogen has 3 isotopes: hydrogen 1, hydrogen 2, and hydrogen 3. What is different about them? Well, they have different numbers of neutrons. Hydrogen 1 has no neutrons in the nucleus. Hydrogen 2 has 1 neutron. Hydrogen 3 has 2 neutrons. Where do we get the number 3? 2 plus 1 is 3. 1 plus 1 is 2. 1 plus 0 is 1. Carbon, for example, we have carbon isotopes. This carbon isotope has 6 protons, 6 neutrons. So this is called carbon-12. Radioactive carbon-14, 6 protons, 8 neutrons. 6 plus 8 is 14. Carbon-14 is radioactive. Hydrogen-3, called tritium, is radioactive. Why is this important? Well, if something was once alive, we could use these chemicals, carbon-14, and comparing it to carbon-12, and we can figure out how old something is. The mass number is this number. Hydrogen 1, that's the mass number. Hydrogen 2, that 2 is the mass number. Hydrogen 3, that 3 is the mass number. Mass number is the protons plus the neutrons. Okay. I mentioned to you already that carbon-14 and hydrogen-3 are basically radioactive. They are unstable. Their nucleus is unstable. What does that mean? It means that they break down over time. We use the term half-life to describe how long it takes to have half of a substance. For example, if you have a bunch of carbon-14, let's say you have 100 grams of carbon-14, it would take 5,730 years to have 50 grams of carbon-14 remaining, which means the carbon-14 broke down over time and became something else. You will no longer have the original amount of 100 grams of carbon-14. In 5,730 years, it would be halved, basically. These are other chemicals and other, elements and their half-lives. So some of them have extremely long half-lives. So what we do, we use carbon dating to tell how old something is that was actually alive at one point. Why was it alive at one point? Because it contains carbon. It is what's called organic. If you remember back, organic substances are hydrocarbons or things that contain carbon, hydrogen, and other elements attached to them. So I just described you what happened. And I have actually a really good picture on this next slide. So for example, you are constantly taking in carbon-14. How do you take carbon-14 into your body? You take it into your body when you eat. The food you eat has carbon in it. So therefore, we are actually ingesting radioactive carbon all the time, every time we eat. All right? At death, you are no longer taking in carbon-14. Therefore, carbon-14 is built up in the tissue of an organism. When this organism dies, it is no longer taking in that carbon-14. Therefore, the carbon-14 will begin to break down. As it breaks down, the ratio of unstable carbon-14 compared to stable carbon-12 begins to change. At 5,730 years, you will have 50% of that original carbon-14 left. Double that number, 11,460 years, you will have a quarter or 25% of the original carbon-14 left. We can use this to calculate roughly, or not roughly, we can calculate basically about how old an object is using carbon dating. Rocks can be dated this way. They are not using carbon. They would be using other radiometric dating elements such as silicon, for example, and looking at their breakdown time. All right, so let's talk. So we're talking about the history, and most people don't understand how life began, and no one understands really truly how life began. But there's some hypotheses on how some of the early building blocks of life could have shown up. O'Parent and Haldane basically said, hey, listen, early Earth had ammonia present in the air, had hydrogen gas, had water, and hydrocarbons such as methane were available. All right? What these guys hypothesize is that these chemicals, these compounds, could probably have formed some of the early organic compounds on Earth that became the building blocks of life, basically. So they hypothesize that energy from ultraviolet light, heat, lightning, you name it, those things came into play, basically. So some scientists named Stanley Miller and Urey said, listen, let's build an experiment testing the O'Parent's hypothesis. What did they do? They took ammonia gas, hydrogen, water, methane. They put them in a container. They applied a spark to simulate lightning. They heated it up. They zapped it with ultraviolet light, lamps, things of that nature. And what did they notice? They basically collected organic molecules, basically. So they actually took all those things, put them into a container, sparked it with electricity, heated it, did all those things, and what actually formed? They found amino acids, the building blocks of proteins, ATP, which is energy, nucleotides, which are the building blocks of DNA and RNA. Does this explain how life began? No, but does it explain how the building blocks of life could have shown up? Yeah, it may explain that. So this is just some history of where the early organic compounds could have come from. Another hypothesis out there, people believe that they're called seeds from space in certain books. What they mean is they think meteorites, rock debris, landed on Earth, and when it landed on Earth, that rock debris brought with it organic molecules, maybe possibly even bacteria and things of that nature. All right. So what were the first cells on Earth? Well, if you remember a second ago, oxygen wasn't in that list of chemicals. O2 oxygen was not present. Therefore, the first organisms on Earth did not like oxygen. Therefore, they were what's called anaerobic. All right. They were probably very, very simple, which means they were probably bacteria. So they were probably prokaryotes. All right. They do have some extremely old fossils. Down here, I'm showing you a 3.4 billion year old fossil in Washington, I believe. It resembles modern bacteria, basically. All right. Chemosynthesis. Well, this is probably one of the early processes that formed what's called our autotrophs. Heterotrophs are like you and me, organisms that cannot produce their own food. We need to ingest food. So what is believed is chemosynthesis probably came first, not photosynthesis. A lot of organisms were probably not living on the surface of the Earth yet because it was a very rough and harsh environment when we're talking 3.5 to 4 billion years ago. They believe organisms probably started producing their own food autotrophically at the bottom of the ocean using chemosynthesis. Chemosynthesis doesn't use doesn't use light, it uses chemicals. It uses sulfur and the energy that's found in the oxidation and reduction of this sulfur compound. When you break it down using oxidation, it can generate energy, and that energy can be used to produce food. And food, in this case, chemosynthesis, is going to produce organic compounds such as sugar. So this would form the base of the food chain at the bottom of the ocean, for example. The organisms that do this are primitive bacteria or bacteria-like organisms, or prokaryotes, excuse me, called archaea. All right. Photosynthesis basically didn't arrive until about 3.5 billion years ago. The previous thought was around 3 to 3.1 billion years ago, but they found fossils recently called stromatolites. And these stromatolite fossils are basically fossilized prokaryotes. And these are pictures of them right here. I believe these ones are found in New Zealand area. This is a very recent find in the dating of the process. Basically, these are basically where these prokaryotes probably died and then became rock structures. So it basically took a long time for oxygen to show up on Earth. Oxygen showed up probably about 1 billion years after other life forms showed up. Okay. Early oxygen didn't last very long. It didn't last very long because it was such a rough environment that it was bombarded by ultraviolet light, and it was breaking it down and forming ozone basically at that time, which is O3. Ozone is a nice thing when it's really high in the atmosphere. We'll learn this in our next chapter. Ozone very high in the atmosphere blocks ultraviolet light and protects us basically. Ozone low in the atmosphere is not good for us. It's actually toxic. The first eukaryotes, well, where did they come from? Well, you probably remember back in Unit 2 when we talked about cells, and we talked about this process called endosymbiosis. Where did these first eukaryotes come from? They believe they evolved from prokaryotes. So what is the idea of the endosymbiosis? It is an idea that smaller prokaryotes, such as smaller heterotrophic prokaryotes or smaller autotrophic prokaryotes like these ones here, they were engulfed or swallowed up by larger prokaryotes. They ended up living inside of these larger prokaryotes, and actually they became, so to speak, friends. They had a mutualistic relationship. They benefited each other. The photosynthetic prokaryotes eventually became what are known as our chloroplasts, and the heterotrophic prokaryotes later on became our mitochondria, or energy producers. So this goes back to Unit 2. You may remember. What's the evidence for this? The evidence is that chloroplasts and mitochondria have a lot of similarities to bacteria. They replicate independently. They have their own DNA. They're found in circular arrangements like prokaryotes. So they have a lot of similarities to prokaryotes. Right? We are officially done with the history of life on Earth. I will set up a second lecture, which will be evolution.